Okay, good morning, everyone, and welcome to the ELIXIR Friday Coffee, which is our series of seminars and lectures devoted to the services and tools the infrastructure is providing. To demonstrate that we are having also a different focus than only the proteins. So we will today be talking about nucleic acid and specifically about RNA. And David Hoksza from Faculty of Mathematics and Physics of the Charles University will tell us about Traveler, which is template-based RNA secondary structure tool for visualization. So David, please, floor is yours.
Thank you. So good morning, everybody. Before I start, let me say that Traveler started actually as a side project or byproduct of RPredictor-DB, a database of predicted at that time only ribosomal RNA, which was a project led by Josef Pádek from the lab of Jiří Vohradsky. And the structures there were, and I guess still are, predicted by CP Predictor tool, which will be presented actually next week on the Friday Coffee. So don't miss it. That's the real deal. Today we will be talking only about how to generate layouts of RNA secondary structure in such a way that the layouts are consistent, reproducible, and recognizable. So let me start with a bit of background and motivation. So as all of you know, I guess RNA secondary structure corresponds to the Watson-Crick base pairs interactions and specific combinations of the base pairs and unpaired nucleotides form so-called secondary structure motifs, such as hairpins consisting of stems and loops, inner loops. Just let me turn on the laser pointer. Yeah. Inner loops, bulges, and so on. So these are the most common ones. And what is important is that some of the motifs or their combinations are functionally important and thus conserved across species. So we would like not only for the motifs in the layout to be discernible, but also to appear at the same position in the layout of different homologous species, which would in turn enable cross-species comparison. So how do we go about laying out these secondary structures? So the problem of RNA secondary structure visualization can be treated actually as a graph drawing problem, but the vertices of the graph corresponds to the nucleotides, to the bases, and edges to the secondary structure, to the base pairing information. And we would like to assign 2D positions to the vertices and polylines or splines to the edges, right? And the graph drawing problem is a very well studied problem in computer science. So there exist many approaches to, I would say, general graph drawing. All of them basically aim at arriving at a layout which would be optimal with respect to some aesthetics criteria, such as number of overlaps and several others. However, these approaches can't be directly applied to RNA because there are some specific requirements which mostly relate to how the motifs, individual motifs, should be laid out. So, for example, for hairpins, they should be laid out like the stem on a straight line, loop on the circle, and so on and so forth. However, these criteria are first only loosely defined, and second, they deal with individual motifs, but there are basically infinitely many possibilities how to position these motifs with respect to each other, right? And that might not be a problem with a small molecule like this, because no matter how you position these motifs with respect to each other relatively, you always will understand what you are looking at, right? But it can be a problem with large molecules, such as, for example, ribosomal RNAs. So where people are used to see, for example, the layout of small ribosomal subunit in a specific way, so which is called standard layout, and they expect to see in the center the conserved core and different features at different positions, and this should be the same across all species, basically, because it's very well conserved. And the layouts, the existing layouts, the desirable layouts, are typically manually or semi-manually created by a domain expert. And if you want to create an algorithm, a program which would create these automatically, you would have to somehow codify the decisions an expert made when he or she was creating layout in order to be able to replicate it, but that's basically impossible. And that's a major issue, as evidenced by the layout achieved by the existing tools, as you can see here. Either the layouts are, so the secondary structure, for example, is, or motifs are not discernible, or it doesn't look as it should at all, right? So we came up with a solution to this problem, which we called, in general, template-based graph drawing. And it's based on the observation that structures of ribosomal RNAs, or also different classes of RNAs, do not differ much between species. And all the expert-drawn layouts are based on the same blueprint, so to say. So the idea was that if we want to lay out a structure for which we do not have such human curated layout, we could use an existing layout of a different but similar structure and tweak it to fit the structure we are interested in, right? So you can imagine that you do not have the layout for a human ribosomal subunit, and you have it for fruit fly, and because they are quite conserved, the secondary structure, I mean, you could tweak this image to arrive at this image. Right? So that's the basic idea, and it's the same idea as for example, prediction of like homology-based predictions of protein structures or RNA structures, for that matter. But it's sort of one level above, in the sense that here you have two structures, template and target structure, and you have also the template layout, and you want to generate the target layout. Right? And that's the basic idea of the algorithm, which is implemented in a Traveler. So specifically how it works is that on the input of Traveler, you need to pass in the template secondary structure and its sequence, and template layout. So that's the one you already know. And then you need to pass in the target secondary structure, and you would like to, and the goal is to generate target layout which would resemble the template layout. Right? So how do we proceed? In the first step, we generate a template, and template and target 3 representations of the secondary structures. Then we create mapping between these two trees, and the mapping tells you which nodes, and nodes corresponds to either base pairs or unpaired nucleotides. So which nodes need to be inserted, deleted, or relabeled to turn the template tree to the target tree, or vice versa, right? So that's the same thing as with sequence edit distance, for example. Yeah. So now when you have this mapping, then we associate this so-called tree edit operations, insert, update, and delete, with layout modification operations, which enables us to transform the template layout into the target layout. So for example, inserting a node at this position requires some layout modification operation at corresponding position in the template. And now the important part is that the residues which need to be moved to make space for the insertions can be located in the subtree of the just inserted node. So then you just apply a translation vector, that's this black arrow, to each of the residues in the subtree, thus moving them in this direction and making space for inserting of the base pairs in this base pair in this case, right? Now for different insertions at different positions or deletions or updates, we do slightly different modification of the layout, but the idea is still the same. So the tree structure informs the layout modification operation, right? The local topology of the tree determines what layout modification operation needs to be done, and that's the whole thing, right? So for example, so I can't give you any like quantitative comparison with other approaches because there are no approaches which would be solving this problem in this way, this template-based drawing, right? So I can show you just an example. So here we are in the situation I described 2 or 3 slides ago. So imagine that we have a template structure and layout, which would be fruit fly, and we have the human RNA secondary structure, and we do not have this layout, right? And this is what we would like to achieve. And when we run Traveler in this situation, it will generate a layout which looks like this. And it should be as close as possible to the template, to the manually curated human-generated layout. And obviously it is. So I'm not sure how well you can see this, but the red residues are inserted, the green ones are relabeled, the blue ones mean that there had to be some more advanced repositioning at that position. But basically, yeah. So the colored residues are those where the target structure differs from the template structure. And the green ones are really easy to do. The red ones and the blue ones are the ones which are challenging. Okay, so Traveler is implemented in a software which is a command line application written in C++, and on its input it takes on its input the target structure. The target structure is a FASTA file which basically looks like this. So you have there one line is the sequence, and first line is the sequence, and the second line is the secondary structure in the dot bracket notation. Then as for template, you need to pass in again the secondary structure in the same format and a template layout. And this we accept 3 formats, 3 types of the layout. So first, PostScript files from CRW, which stands for Comparative RNA Website or Web Server, not sure. So there you have the layouts for example the ribosomal subunits, but also other classes. Then the second type of input is the Varna SVG file. So Varna is probably the most popular tool for manually drawing secondary structure of RNAs, and it can output an SVG file which Traveler is able to parse. So you can draw your own template in Varna, Or if you have the template in completely different format, you can write your own parser and convert that format into an XML, which then Traveler can process. And as for output, it outputs an SVG file, which can be then exported to PDF or PNG or whatever you like, and also XML file. And so the idea is that if you would like to use it then in a, for example, pipeline, then you can parse this XML file to get information about the positions of the bases. Now we also support custom target template sequence alignment, and what this means is that here I was saying that we do this mapping between these trees. For that we use tree-edit distance. And the problem with tree-edit distance is that when the target and template are dissimilar, really dissimilar, it can happen that the tree-edit distance maps like wrong parts of the tree onto each other. And that's really a problem because then the layout is really completely wrong. And the reason is that this tree-edit distance is global, it's not local. So what you can actually do if you know the mapping, so either you can pass this tree mapping, but you can also pass in the mapping on the sequence level, and we then turn it to tree mapping, because we need the trees to devise the layout modification. Sorry. Yeah. Okay, and I said that it's like written in C++, so in order to run it, you need to compile it and so on. That might be difficult for some users because of all the dependencies and so on. So it's also available via a Docker container at this address. Okay, so that's the Traveler tool itself. So Traveler is actually integrated within RNA Central. So RNA Central is probably the largest repository. Like it integrates many member databases, and therefore it's one of probably the largest repository of RNA-related information. And when you go to RNA Central, you can see some secondary structure layouts, and these are all generated by Traveler. So actually, currently, in the newest version, I think there are like 20 million RNA secondary structures, and their layouts are generated by Traveler. But I should emphasize that these secondary structures are not experimentally determined from the structure, but they are predicted. And for the prediction of the secondary structures, which are in RNA Central, there exists a newly developed pipeline, which is called R2DT, which stands for RNA 2D templates. And at the end of the pipeline sits Traveler. And the pipeline works as follows. So you pass in your input sequence for which you would like to get the layout, the secondary structure and the layout. Then behind R2DT is quite extensive library of templates. So currently over 3,500 templates. Then most suitable template is selected via RiboVor or tRNA scans. So RiboVor is a tool developed at NCBI, and tRNA scan in LoveLab. Then this input sequence together with the template is passed into Infernal, which is another tool, like co-evolutionary based tool, which folds the sequence into the secondary structure based on the template. But here I should really emphasize that what Infernal does is only like sort of copying over the base pairs from the template, but it does not add additional base pairs, meaning that you should be careful when interpreting the resulting secondary structure, because if you see a base pair in the secondary structure, predicted secondary structure, that is probably correct. But if you do not see a base pair, that might mean that it was just not added there, because R2D does not do any effort to add base pairs which are not in the it does not see in the template, right? So that is work to be done. Anyway, then you get your secondary structure. You also have information about the best template, because there's a template using which the secondary structure was predicted, and then you can run Traveler. And here we actually use this possibility to pass in the target template sequence alignment, because you were using this template, so you know the really correct alignment, right? And that's why it was actually implemented into Traveler, and it really greatly improved the layouts especially for a large RNA structure, where the tree mapping was often wrong. As for the template library, which I was mentioning here, here you can see the statistics. It covers like a wide range of RNA classes. Most of the templates, or many of the templates, are manually curated. For the classes for which we do not have manually curated templates, we use RFEM as the source, but yeah, those are not like very nice. Yeah, because I mean those are classes of RNA molecules for which there is not like community agreed upon standard layout, I would say, or at least we do not have it. And as for R2DT, actually you can run it either as a command line app, and there is now running this from command line would be very difficult because there are a lot of tools involved. So there is again Docker container, and there is a lot of documentation at this address, so you can run it from command line from there. The easiest way how to use R2DT, and actually also Traveler probably, is to use this web server, R2DT web server, where you can pass in your input sequence, let R2DT automatically select the best template, or you can, if you know better, you can pick the template and let it do its job. It predicts the secondary structure and uses Traveler to lay it out. Also, there is an API which you can use to do the same thing. And I said that you can select your template from the library, but you can't pass your own template. But if you wanted to add, for example, if you are expert in some types of RNA for which you believe the standard layout should look like something, there is a way how to add new templates into R2DT. So then everybody out there will have access to the template which you will provide within the R2DT pipeline, I mean. And yes, that's it. So if you have any questions, I would be happy to answer them.
Yeah, David, thank you very much for the lecture. We have a few questions, so I'll try to summarize some of them, because supposedly Josef Pánek will tell us next Friday Coffee about the template-based prediction. So the question number one is, can you classify RNA secondary structure in a similar way how, for example, the CATH does for proteins? It means if you can actually assign classes, families, or architectures.
That's what RFEM does, right? In a sense. So RFEM is doing classification, but they are not like the ones in Catch where you have the architecture basically of RNA, right?
Okay.
So yeah. Sorry, go ahead.
Okay, Karel is asking the question, is there a possibility to generate automatically the consensus template given a set of similar RNAs, for example, RNA for S protein in SARS, MERS, SARS-2 or AMR in vaccines?
Okay. Currently, no. No. I mean, all the templates which we use are like manually generated, and so the point of this is that there is community agreed upon standard layout, right? So if you generate something automatically, it's surely not community agreed upon standard. And so that's one of the reasons. Second, it would be pretty difficult to do this automatically, although it's quite a nice challenge. And yes, it's a nice idea. I think I will talk to Karel about this, and we can think about something.
Okay. Here, guys, raising a question: if using only the so-called secondary structure, when you actually take into account only a Watson-Crick pairing, you are missing a huge portion of contact. So 3D modeling is probably unreliable. So yeah.
So we also take into account like pseudonotted structures, just that for the mapping and generation of the layout, we take only the Watson-Crick base pairs. So how do we go about it? That at the beginning we leave out all the other types of pairs. We generate the layout based on the Watson-Crick base pairs, and then we reinsert those into the final layout. So so the orientation of the layout is determined by Watson-Crick base pairs, but then we add the rest of the structure. Because I mean the other types of bonds go like across, like really across the layout, and it wouldn't be wise to do the layout based on them.
Okay. So Karel is asking if can travel use Leontis-Westhoff nomenclature for RNA contacts?
No, currently not. We are not dealing with that right now.
Okay, so I think this is the last question we put here. So David, thank you again very much, and see you next Friday on CP Predictor, which will be template-based prediction of RNA. Thank you very much for your attention, and have a good weekend. Bye.